when we last left off, and how that these, an example we were going to use was glycolysis and gluconeogenesis, and how they were not fetal cycles, and that there were different ways to go about regulating them. We talked about allosteric control, covalent control, substrate cycling, and at the genetic response level. And those were just some of the examples. And I do expect you to be able to give specific examples for each of those. And let's see. Oops. And so first up, we had discussed hexokinase. In particular, how hexokinase, there were different isoforms of this enzyme that 1 through 3 were particularly in the muscles, but the liver one had a very specific isoform called hexokinase 4. Many times it's also called glucokinase, and how it had a much lower affinity for the glucose. And so we talked about what happened in the glucose and the reasons why, and what happened in the liver with respect to this process, and reasons why it would be important that it would have a much lower affinity for its own substrate than the muscle forms. And so what was some of the reasons why it would be important for the liver to have a lower affinity than, say, the muscles or the brain or anything like that?
They need it first.
Because it doesn't want to take away from the organs like the heart, the brain, things like that, and it undergoes gluconeogenesis. Okay, so therefore, it needs to have a lower affinity for it, so it doesn't compete against gluconeogenesis within itself. But it also needs a lower affinity than the other forms in the other tissues, so that way it doesn't compete with those. Okay, and then finally, we also talked about how hexokinase 4 and glucose 6-phosphatase were transcriptionally regulated, for the longer term, or that's a longer term response rate than, say, allosteric control or covalent control. Now, I don't know, did we get to this figure? I wasn't sure if we got this far yet. I didn't write it down where I left off. Okay, so this is, just a you can think of it as a type of, similar to Michaelis-Menten, except the fact that this hexokinase 4 in particular doesn't undergo Michaelis-Menten kinetics in the classical sense, but it's, relative velocity over per substrate concentration. So first off, let's just discuss about what does it indicate with respect to hexokinase 1? What kind of curve is that? It's hyperbolic. It's much more hyperbolic than hexokinase 4. And once again, hexokinase 1 is in what kind of tissue? Which cell tissues would have hexokinase 1 in particular? Like we're talking like the muscles and things, versus hexokinase 4, which is in the liver. Okay, so that's why I would expect you to be able to talk about, whoops, let me bring it on down. Come on down. There's like a lag, at least on my computer. All right, talk about, for example, when the blood, this is the glucose level, the serum, like it has frozen. I'm not sure why the internet is going down. When the serum glucose level gets low, relatively low, say it's at 5 millimolar, you can see there's a lower affinity and lower activity in the liver versus, well, I'm pointing at the hexokinase 1, there we go, versus the same concentration in the muscles, which makes sense. Say, it's been a while since you've eaten, so therefore you don't need the liver should not be competing with the muscles, the brain, other tissues for the sugar. Now, if you've been eating, so it's at a higher concentration, now you can see that the activity starts to increase. We've still saturated the one for the muscles, which, come on now, there we go. We've saturated the one for the muscles, but the activity is still going to be lower, and that's going to be true essentially at all concentrations for glucose, that there's a lower activity within the liver for glycolysis, that's for glycolysis with respect to the liver, than those that are in the other tissues. All right. So glucose is produced via gluconeogenesis in the liver whenever your serum concentration for glucose is low, which that should make sense, okay? And it's much less likely to get trapped, to go back into glycolysis. So what that means is, let's say for example, see if I can get this to work. There we go. Is that if you just think about it, glycolysis, which I'm just going to put it this way, goes this direction from glucose to pyruvate. When your blood serum level, let's get a different color. Let's say that if this is the concentration of your blood serum level is decreasing. So then what's going to happen is you're going to get this kick in to go from pyruvate to make glucose in the liver. But you don't want your liver to say, okay, hey, wait a minute, there's glucose that's now here in the same cell that I'm using, and so to turn around and have it to go right back into make pyruvate, that would be futile. And so what happens is, since the serum level of glucose is low, it's not above its km. It's well below its km, and so it'll go ahead. It makes glucose using gluconeogenesis. So this would be GNEO. Makes glucose, and then that glucose can then go out into the blood. Okay, and then that will start to cause the concentration to rise up. Does that make sense? So that way then the blood glucose level is higher. It'll then go to the brain, to the muscles that need it, until you eat or until it gets too high, and then it can go back, and the liver can then take over. And if the liver needs additional energy, it can go through glycolysis then. If it's fine energetically, it can then even store it as glycogen, which we'll talk about later. Let's see if I can get this now to... No, I don't want to export it. Did it work? Okay, awesome. All right, so that's what I was talking about with respect to that. Now, hexokinases 1 through 3 are not inhibited by glucose 6-phosphate. I'm sorry, they are inhibited by glucose 6-phosphate. So that means it's inhibited by its own product. However, hexokinase 4 is not. Okay, so that way you don't have to worry, or the liver doesn't have to worry about being concerned about being inhibited by its own product, since it only kicks in to go through glycolysis at much higher concentrations of glucose overall. Now, hexokinase 4, which is the one in the liver, is inhibited by a regulatory protein. And so that's why this is what helps dictate whether or not the concentration is getting high and the liver no longer needs energy, or that the liver does need energy. So if the internal concentration of glucose reaches a certain level, like you've just eaten a carb-heavy meal, right, then the glucose will bind to this complex to release hexokinase 4 to initiate glycolysis, because maybe the liver needs energy. Now, if it gets too low, like during a fast, then hexokinase 4 is allosterically affected to bind to the regulatory protein to inhibit glycolysis. So why does that make sense that during a fast that you're going to want the glycolysis in the liver to be inhibited? So it's between meals.
To conserve energy?
You want to conserve the energy for the brain and for the other tissues. Okay. All right, so this is a little cartoon to, well, I don't know if there's a big delay in there or not. There's a little cartoon here to show what this means. So let me see if I can try to... I wish this had a better pointer, because the pointer screen is really tiny. So what happens here is, first off, this GLUT2, that's a glucose transporter 2, it's really... We've talked in the past about some of those transporters, like the sodium potassium ATPase one, where you have to keep a much higher concentration of one ion on one side versus the other. It's not that way with GLUT2. GLUT2, it's pretty free-flowing with respect to glucose, to where for the liver cell, the internal glucose concentration of the liver cell is essentially the same as that that's inside your blood. Okay, because that's how the liver can kind of tell what's going on, like what the relative concentration is. Okay, so whatever the concentration is here, is essentially what it's going to be in the inside and the cytosol, the hepatocyte, because of this GLUT2 transporter. So what happens here is that whenever the glucose gets, oh, come on now, it's frozen once again. There we go. Whenever the glucose gets high, it's going to turn on this process of the hexokinase 4 attached to its regulatory protein inside the nucleus. It's going to cause it to come out, and it's going to lose its regulatory protein. Okay. So first off, why is it important that the one that's attached to the regulatory protein is in the nucleus versus being in the cytosol? What happens in the cytosol that's not happening in the nucleus, or that doesn't happen in the nucleus? Glycolysis. We always have to think back to where location. Glycolysis is in the cytosol versus being in mitochondria or the nucleus. Okay, so glucose that is high in the serum, so then it comes and it gets to the point where it can come out here in the cytosol, undergo glycolysis for the liver. Now, if it gets to a point where glucose 6-phosphate doesn't inhibit, but much further down is fructose 6-phosphate. If this starts to build up, it's going to build up because the liver has plenty of energy now. Okay, and so if the liver has plenty of energy, it goes and it actually turns on the process to cause the regulatory protein to bind to hexokinase 4. It gets transported into the nucleus, and so therefore we can undergo glycogen synthesis. Oh, I gave it away. We can undergo glycogen synthesis. You can either do gluconeogenesis or you can do glycogen synthesis because the liver no longer needs its own energy. Whoops. Yep.
The regular protein bind and then it goes to the synthase?
Well, I'm not for sure which happens first.
Okay.
Okay, it just causes that process to occur. All right. So that was we talked about how step 1 was regulated, the hexokinase versus glucose 6-phosphatase. Now we're going to talk about PFK1 versus fructose 1,6-bisphosphatase. Remember, that's step 3 in glycolysis, and I don't remember the numbers for the gluconeogenesis, but what would be the antagonistic gluconeogenesis enzyme. They are controlled via allosteric, covalent, and substrate cycling mechanisms, and it's reciprocally regulated, which makes sense. When one's inhibited, the other one's activated. When one's activated, the other one's inhibited. What now? Okay. So, PFK1 is really important because it's called the concomitant enzyme. There's a big vocabulary word for you. It's the step that says, okay, from this point on, you're going to go towards glycolysis. You're committed. Okay. Hexokinase 1 doesn't necessarily commit you to glycolysis. You can cut back and go backwards. All right, but once you get past step 3, onward Christian soldier. Faith integration. Faith integration there. So, yes, you're not going to go on to do the other roles because we're past the glucose 6-phosphate, because another possibility was the pentose phosphate pathway before. But you can't do that. You're going on towards glycolysis. So besides binding the substrate, it has many, many regulatory binding sites. Okay, so now let's just think about some of the possible things that could activate it or inhibit it, because some of them would make perfectly logical sense. They're the usual suspects, and some of them would be kind of new. So what are some things that could what would make sense? What's made, or further down the stream, so to speak, in glycolysis, or because of glycolysis, that would make sense that it could possibly inhibit PFK1. Pardon? What did you say? ATP. That does. I mean, that makes great sense. Come on now. ATP. All right. What's something that you think that would make sense that could activate it? That if you're low, oh, I forgot that it changes the color that up there, so it didn't come out very dark. What makes sense that could activate it? Like if you get low with it, then it makes sense. Okay, we're gonna need to go on with glycolysis. This doesn't inhibit it, activates it. Turn it on. ADP. Okay, and we're gonna find out AMP as well. But ADP and AMP. So those make perfectly logical sense because those are the hydrolysis products when ATP gets used. And then before I go any further, I just want to point out that this one's also related via phosphorylation because I should have put it on a different slide, but I didn't. Okay. So here we go. Once again, these are the kind of questions or things that I want you to pick up on the logic. You know, what does this mean? Be able to read this. That's PFK1 activity, so it's glycolysis. Make sure you know what pathway we're talking about. Fructose 6-phosphate. Make sure you know, is that a substrate? You know, is that the product? What is fructose 6-phosphate with respect to PFK1? Is it the substrate of the product or the activator, inhibitor? What is it? It's the substrate. So essentially this would be similar to what you'd see with Michaelis-Menten. Okay, so first of all, what does this shape mean?
Allosteric.
It's allosteric. And you can just qualitatively look at this and discuss about the relative allosteric. When ATP, when ATP increases, what happens to the allosteric? Does it become more allosteric or less allosteric?
As the ATP rises?
As the concentration of ATP rises, it becomes more because it becomes even more sigmoidal looking, more curvy. Okay? So is ATP an activator or an inhibitor? It's an inhibitor because you have to get to. Don't do that. I hit the wrong one. If we get to, let's just pick a constant. If I can get the arrow to go down, it's frozen once again. There we go. If you get to any particular fructose 6 or substrate concentration, in the presence of high ATP, there's not much activity. Versus if there's not very much ATP around, you get a much higher activity. So that's how you can say, okay, this is clearly an inhibitor. Whereas you would expect a different effect of that if we were looking at ADP, since we said that ADP was an activator. I love this next figure, by the way, because even though this one you can't really read in the one box. We've already talked about this. What are the significances of ADP, AMP, and ATP? Why does it make sense that ATP inhibits versus AMP, ADP activates? But the next two are the ones that are new. Okay? And I didn't check the page numbers, so the page numbers may be page numbers may very well be incorrect on this one, but okay. So first off, let's go first. Citrate, good, makes sense. First off is citrate. So we have to talk about from where did this originate and why is that important? And then we'll talk about fructose 2,6-bisphosphate. From where did that originate and why is it important? So it's 2,6, not 1,6. So it's not the direct product. It's something else. Okay? That's why it's really important where the phosphate is located. So if we just focus on the citrate, where does it make sense that the citrate normally is? Citric acid cycle, which is also called Krebs cycle, which is also called, there's another one, the TCA cycle. What does TCA mean? It's a tricarboxylic acid cycle. And the reason why is because citrate's tricarboxylic acid, but so are some of the other intermediates, or metabolites, I should say. And so that's why all three of those mean the exact same thing. That and they used to talk about the Krebs bicycle, because it's citric acid cycle and urea cycle are intertwined. Okay. So first up is, before I go any further, those page numbers may be wrong because I forgot to check the most recent edition. But there is a section on there that talks about the role of ATP and ADP and AMP. I mean, you should just understand we've already talked about that before, and so you just want to review that on your own. But like I said, it may not be 575 to 577 because I didn't write down what edition I took that out of. Okay, so citrate, which is just the ionized or the carboxylate form of citric acid. Citric acid comes from the citric acid cycle or the Krebs cycle or the TCA cycle, as we mentioned, which is part of the aerobic oxidation. We talk about anaerobic versus aerobic. And so pyruvate, we're going to find out pyruvate doesn't go directly into the citric acid cycle. It's one of the Moses in the Holy Land kind of ideas. But pyruvate becomes something else, and that intermediate can enter in and enter the citric acid cycle. We're going to find out that citrate is just a good indicator of when your cell has plenty of energy. And so, because its concentration tends to accumulate whenever fats and proteins have been oxidized. And so it's one of the metabolites of the citric acid cycle that can start to build up. So that's why it does make sense that it's going to inhibit glycolysis and activate gluconeogenesis, but it's just you don't find out until I think it's the next chapter about the citric acid cycle. And once again, it's called the TCA cycle as well because if you look down here, there are 3 carboxylic acids. Now we're going to spend some time on fructose, quite a bit of time with respect to the fructose 2,6 bisphosphate and how it regulates, because then we get to talk about glucagon versus insulin, which every diabetic should know something about, hopefully. Okay, so this was just a little picture showing everything so far. I mean, it's just, instead of it being vertical, instead of being horizontal, it's vertical, and it clearly says gluconeogenesis versus glycolysis, and so on your own you can look at that. It comes directly from your book, and this should be, I'll try to make this available later today, because we'll get through most of this chapter. Okay, so this is fructose 2,6 bisphosphate. It regulates both gluconeogenesis and glycolysis. It has to do with the glucagon and the role of glucagon and the enzymes that are involved in making fructose 2,6 bisphosphate, which I'm just abbreviated as F26BP. And I like to put the comma in, but your book did not. So it's not 26, it's 2,6. Okay. Now these, and this is what it looks like, but you should, if I give you what fructose looks like, then I would expect you to be able to know where the phosphates were. 1,6 versus 2,6. You just have to realize that, let me see if I can get it over there. Come on now. Oh, here, it's easier if I just write. Well, I said that. No, it's not going to. Okay, but you just have to remember that carbon number 1 is the one that's down. Because some people just always assume it's the one next to the oxygen, but remember it's not. If you have to go back to last semester, the scorpion's tail had attacked it, or the carbon, the nucleophile attacked the second carbon, not the first carbon here. Since this is a furanose, not a pyranose. Okay, let me erase that. If I blow this up, will it blow up? No, never mind. Okay, so I would expect you to be able to talk about qualitatively what these figures mean. Like, once again, allosteric, does it inhibit, does it activate? So if we look, and which enzyme is which? So first off, if we look at PFK1, is that glycolysis or is that gluconeogenesis? That's glycolysis. So does F26BP activate it or does it inhibit it? It activates. See, you add it and it goes up at that concentration. This is the substrate. That's the substrate. Fructose 6-phosphate is the substrate for PFK1. Whereas if you don't have fructose 2,6-bisphosphate around, then if it's not around at all, then the activity is much, much less. Okay? So glycolysis gets turned on in the presence of F26BP. Whereas gluconeogenesis, we have FBPase1 here, which, since I've already asked you what the other one was, you would know that this one is gluconeogenesis. Fructose, whoa, no, don't do that to me. Fructose 1,6-bisphosphate is its substrate. And here, I mean, if you look at the same thing, it inhibits it. So you can see that the relative KMs, that's what they're showing here, is the relative KM, or I shouldn't say KM, it's the relative K1 half, where the concentration of it, or KI, it's a better one. KI is really, really low. That really is more of a KM there, but the KI here is much increased. Or the amount of the relative Km there in the presence of fructose 2,6-bisphosphate is really high. That's probably the better way of saying it. Okay. So I mentioned that it has a role in glucagon. What does glucagon do? Pardon? Right. What is it? First of all, it's a hormone. So, and it's the antagonist of insulin. Okay. And so it's what signals for the liver to release glucose. So it should make sense that glucagon would say, okay, we only have two possible ways to get glucose. One is if you've got plenty of glycogen stored, glycogen will start to break down and release glucose. That's an easy way. Another way is through gluconeogenesis. Whether you're using pyruvate, which is the obvious one, lactate, which, does anyone know what enzyme it is that helps go from lactate to pyruvate? Which someone probably, I think she knows. I think that we're going to discuss it in class in a couple of hours. Lactate dehydrogenase. Okay. Glycerol and certain amino acids, which we really haven't discussed that, but glycerol we can. Later on we'll talk about how you can break down glycerol to get to glucose, and then certain amino acids are glucogenic. We've already talked about that. All right. Now, if blood glucose is too high, then that's where the insulin kicks in, and believe me, we'll go into great details with insulin, how insulin is regulated during the hormone regulation chapter. Okay. So it tells the liver you need to use glucose for fuel. We've got plenty. You can store the glycogen. All right. And so one way that we can do this really fast, and the effects of these hormones are so fast, is through the use of fructose 2,6-bisphosphate. And I just showed you on the graph how it greatly alters the Km for the substrate by activating PFK1 and inhibiting FBPase1. And then you have to really go back to the end of last semester because it stimulates adenylyl cyclase of the liver. What does adenylyl cyclase do? Cyclases make cyclic, and so adenylyl cyclic will make cyclic AMP. And what is cyclic AMP? What does it do? It's a signaling molecule. Remember, it's a secondary messenger. It goes on and it turns on what enzyme in particular? Cyclic AMP turns on very, very important enzyme. What? Pardon? It's a very generic name, but it's a very, very important enzyme that's used because it's the next step in that cascade that turns on and turns off a whole host of enzymes. Okay, but what one? It's one enzyme in particular that cyclic AMP activates. It's a kinase. Cyclic AMP.
Epinephrine?
No, epinephrine's not a kinase. Epinephrine's another hormone that ultimately turns on cyclic AMP. Cyclic AMP.
Protein kinase A.
Protein kinase A, PKA, AMP. Cyclic AMP.
Oh my goodness.
Cyclic
Okay, that one.
AMP. Yeah, don't forget PKA. Just like PKC was turned on by the C was for.
See, wait.
There's only one secondary messenger that you can probably think of. Well, there's two, but there's one
I'm
that.
done. What?
I remember.
Yeah, I figured Hershey would get this one because he's neurochemistry. He's brought it up multiple times.
Calcium.
Calcium. Thank you. You're not even in that class.
Missed observation.
Okay. The one that's more difficult is PKB, which has to do with the inositol signaling pathway. So, yeah, you'll definitely want to make sure that you review some of those from the signaling cascades. All right, but here's just a little cartoon that just talks about everything that we've been discussing. Epinephrine, which we just discussed as well, which now not let me point to it. Epinephrine is another hormone which has very similar effects, but it's just under different conditions. You know, it's a fight or flight. And so you can see the effects that happen in the muscle versus the liver, but on your own you can go ahead and look at that. But ultimately, the purpose for glucagon is you want to create more of glucose 6-phosphate, which can then ultimately go on to form blood glucose to increase your blood glucose levels. Okay, so yes, please review that. Okay, I'm going to try to get to the next two slides in the next 4 minutes before problem stats overtakes us. Now we have to use our enzyme intuition here. We have, this is the antagonistic, like the regulation of how fructose 2,6-bisphosphate is made. So fructose 6-phosphate becomes fructose 2,6-bisphosphate at the expense of an ATP. So what class or classes of enzymes could that possibly be? Kinase. Have we ever seen anything like this before? Where fructose 6-phosphate becomes a fructose something something bisphosphate.
Yes.
Yes. What enzyme was that? Phosphofructokinase 1. So, using your logic, what class or classes of enzyme, what do you think this enzyme is?
Two?
Oh, yes. Brooke gets a what? So, now, in reverse, now we want to get rid of the fructose 2,6-bisphosphate, and it goes back to form fructose 6-phosphate. What class or classes of enzymes could this be? A phosphatase, and have we ever seen anything like this before where we had a bisphosphate becomes a phosphate?
Fructose 2,6-bisphosphate.
Fructose, oh, you are really, really close. What was the enzyme called in gluconeogenesis?
Fructose 6-bisphosphate?
No, I mean they abbreviated as FBPase 2 versus No, it is it's a fructose bisphosphatase 2, fructose bisphosphatase 1. I would have taken I can't even say it. Fructose 2,6-bisphosphatase is great as well. It's just if you use the abbreviations, it's 2 versus 1. But fructose 1,6-bisphosphatase or fructose bisphosphatase 1 versus fructose bisphosphatase 2 or fructose 2,6-bisphosphatase. That's a mouthful. Okay, and then we'll talk about, there's only a couple more slides for the entire chapter. Oh, no, I guess there's more than that, because I do talk about glycogen breakdown this chapter as well. But we'll finish that up on Monday. So I'll go ahead and I'll end class with prayer.